Hello, everybody, and welcome to this very interesting presentation with Elicera Therapeutics. First of all, some practical issues. You can post the chat. If you have any questions for Dishu, you can post your questions in the chat down right, and I will give the questions to Dishu during the presentation. Then I would like to, of course, welcome you, but also welcome Dishu for this very interesting 20 minutes. This morning, Elicera announced interesting data on their technology platform, iTank. Please go on, Dishu.
Yeah, thank you very much to have me here and talk about our iTank technology. So I will start to briefly introduce our immune system, especially the T cells in our body. So the T cells, actually, you can see they are soldiers in our body. And then they can bear, for example, their receptors expressed on their surface that recognize any infected cells, that is non-self. So they recognize the non-self invaders and then kill them. But however, to battle cancer, which is derived from the self tissue, how we can do that? So researchers have past the 10 years developed a technology called CAR T cell therapy, which in short for chimeric antigen receptor. So the CAR is consist of partially antibody for recognize, and then partially the T cell receptor signaling domain. So in short, the cells can then be engineered to recognize something expressed on the cancer surface and then kill the cancer cells. So then we sort of can redirect cells recognition of the T cells. After 10 years development, there's now 5 drugs of CAR T cell based therapy have been approved, mainly targeting B cell lymphoma and the multiple myeloma. The success of that, for me, I see that 2 reasons. One is that the cancer cells are mainly in the circulation system, so that's why the T cells is easier to see the B cells. And the other one is that they all express this CD19, so it's easier for the T cells to be engineered to recognize the target. However, the challenge to battle the solid tumors, there's a lot of different barriers to block the CAR T cells. For example, the environment, since there's a solid tumor, it's a suppressive immune response, and then there's a dysregulated metabolism, for example. However, we are tackling the target heterogeneity. What does that mean? So here, for example, not all the tumor cells that express the X, the target that you can find. In this case, for example, you can see that the gray tumors, you have this target X, and then you can wipe them away using CAR T cells. However, since there's not all the tumors express this, these red cells will outgrow and form a new tumor that we call antigen escape. So Elicera, in that case, developed a new technology called iTank platform. So if you see, this is a conventional CAR T cells. They kill the tumor cells by recognition, CAR and the target. And Elicera, we call it CAR-NAP T cells. Based on the conventional CAR T cells, we let the T cells to further secrete a protein called NAP. And we believe that NAP will do some fancy work. So why is NAP? Because NAP is a strong immune stimulator. It can induce bystander immunity. And then there's a lot of publication supporting this idea, including our previous publications that, for example, NAP can induce Th1, reach tumor environment, and activate the DC. But anyhow, so our solution that can let our own body to find this X, this target, and due to the bystander activation, we also let our own body to induce the bystander immunity. So that's why we can target even more X and Y. That's the mutations in the tumor. Then we don't need to find exactly what the X is, and our body, our T cells will find it for ourselves. To back up what I say, we did a lot of experiment. I just show you some data here. For example, we evaluated our iTank technology CAR T cells in 5 different tumor models, And then here we have different tumors, for example, colon cancer, lymphoma, pancreatic cancer, and the neuroblastoma. And they are from different mouse. You have black mice, white mice, and this is another type of white mice. And even here we have different CAR targets, either PSCA, CD19, or GD2. So regardless of the mice or target or cancer type, the amazing thing is that you see only these CAR-NAP T cells significantly prolong the mice survival after treatment. However, the conventional CAR T cells doesn't do anything. This is in the solid tumor case, of course. And then in the lymphoma case, yes, you can see that the conventional CAR T also can do something compared to the control. Then, as I said, that iTank can induce bystander immunity and let our body to find the XYZ, right? Here, I show you some proof of that, is that we established a mixed tumor. Here, the red or green, that means you have a target CD19 or you don't have a target the negative one. And then we treat the mice with CD19 CAR T cells. So only kills the red cells. However, when these tumors are established and treated with only the CAR T cells targeting CD19, you can see that only the NARP CAR T can inhibit tumor growth and again prolong the mice survival. And all these survived mice, if you give again the green cells that don't have the target, they can still be protected. That's indicate that we have generated our body to recognize these negative cells. But you should remember that we are only targeting the red one. That means we generate a kind of bystander immunity that recognize something else. And to prove that, we again take out the T cells from these treated mice, and then to read out the interferon gamma. This is a readout. And you can see only the iTank platform engineered T cells here, NAP CAR T, can recognize these green cells. So the take-home message in short is that we generated a good platform that armed the CAR T cells, and then it can induce a bystander immunity. And based on this, and we are discussing, preparing a, sorry for that, we are preparing our first application based on the CAR T cell platform. That's our drug candidate ERC 301, targeting lymphoma. And you can see again, we proved that the lymphoma can be treated with CAR-NAP T cells, inhibit significantly the tumor growth, and also prolong the tumor survival. And this is just show you that before and after CAR T treatment, how the cells will look like. All the tumor cells are dead. And this is also that the quantification of that. You can see that the gray is the tumor cells left over, and the red is after CAR T cell treatment. There's no tumor cells left over.
So this picture you just showed, you still have some green cells left on the after.
Yes. Here I should mention that this is the time dependent, and these green cells, you see that the green is very, very strong compared to the other dot. That means they are under apoptosis already. So if you leave for a long time, the signal will be gone.
Okay.
But then I will show you a completely black picture. It's nothing.
No, that's right.
Yes. So no, these cells are like condensed. So that's why the signal is very bright, and then they are undergoing dying, sort of dying cells. Yes. Okay, as a conclusion, I would just present you the mode of action of our CAR T cells. So our CAR-NAP T cells, iTank engineered CAR T cells, that they first kill the tumor cells by recognition, the antigen, and then on top of that, NAP is released, recruit a lot of other cells. These innate immune cells go and start to kill other tumor cells, not dependent on this recognition. And these tumor cells will be again phagocytosed by DC, and you start a series of immune response, educated our own body T cells. And these T cells then go back to kill, for example, here the purple yellow cells that is not recognized by the CAR T cells. So from this way, we kind of target the tumor from different angle and then try to eliminate a solid tumor. And in the end, I will say that this is not only me did the project, it's a collaboration with a teamwork and also a collaborator from the Uppsala academic who's at Gunilla Imblad's lab. And also our funding agency, especially Elicera, and the mice, of course, very important. Thank you for your time and thank you for listening.
Yeah. Well, then we open up for questions, and I can maybe post the first one issue. So now you today sent out a press release, but will you also release something in Lancet or medical journal about this? Because as I understand it, this is very interesting information in respect of CAR T cells. You took a very big step forward, not only for yourself, but also for other projects in other companies.
Yes, exactly. As you can see here, I don't know if it's still on the sharing. As you can see here,
Yeah.
we tested different tumor, different CAR targets, and then on different mice that mimic different people, for example.
Yes.
And you can see that the technology is not restricted to our own product. It can be universally applied to any kind of CAR T cells, even allogenic CAR T cells. That is also very hot topic, definitely. And regarding your first question, yes, we are finalizing the manuscript that will even I saw it's unpublished, but it's under revision.
Yes.
Sorry, I cannot tell you the journal name, but anyway, I can say that we get quite a good review response. So they like our study, and then there's small modifications. For example, they want us to adjust.
Okay. No, thanks a lot. No, of course. And then a second question, and maybe it's more, maybe it's more for the CEO Jamal, but I ask you anyway. So this open up for some kind of partnerships. I could imagine, and also this will, as you mentioned on your slide here, for CAR T cells, this is some kind of groundbreaking, as I understand it. This will open up for partnerships, and how long should we wait for that, do you think? So how far are you in the process now? You release data, and you have
Because we have our we have our own leading drug using this technology, and then
Yes.
we have two drug candidates, one targeting lymphoma, one targeting glioblastoma, under development. However, we are open for partnership and then licensing out the technology, and the technology is already done. I mean, we are already applying this technology and start to make a virus for GMP virus with BioNTech actually, and then we are already starting the process. Yeah, I would say that next year, mid of next year, we will get the virus. So the technology is kind of matured, I would say, yes, to be licensed out.
Thanks a lot. And then there's a question coming here, very good question. How do you see risk of antibodies against NAP?
Yeah, is that one of the reviewers asking that question as well? Are you the reviewer? So definitely we know that. So antibody against the NAP, yes, maybe there is something like that. However, we see this as a good or bad thing that we can only evaluate in our body, in human body, with a fine dose escalation clinical trial. So in short, antibody against NAP. However, NAP is inside the tumor area, released locally in the tumor area. So the antibody will make the tumor area hot. So from that perspective, I see that this is a good thing. The second thing is that to generate antibody, it takes time. So and our idea is that the NAP will finish all the job during the time, 1 week, that usually you generate anti-NAP antibody. And then during the 1 week, you already stimulate our own body's T cell response. So you already get the T cell target X and Y, Z. The second is that people might already been infected with Helicobacter pylori and then had already existing NAP antibody. So we actually have mouse data I didn't show here, showing that if you give the mouse antibody and then treat it with our NAP CAR T cells, we still get a similar result. So that means NAP can still work even though there's an antibody against it. Yeah. So that's my answer.
I hope you answered the question. I feel sure you did. There's no more questions right now. I don't know if anybody else have a question to Tissue and these very good results. If that's not the case, I would like to thank everybody for listening in to Tissue, telling about their very good results and the iTank platform. Thanks a lot, and thanks a lot to you, Tissue, for presenting in a good way.
Thank you very much.
Have a very good day, all of you. Thanks a lot.
Yeah, you are welcome to send me email or Yamal email if you have any other follow-up questions.
Thanks a lot.
Thank you very much. Thank you.
You're welcome.
Have a nice day. Bye-bye.
You too. Bye.
Bye-bye.